I'm going to start recording now. All right. Welcome, everyone. We're going to get started shortly. Just give a second for everyone to join. All right, I think we are ready to go. Welcome everyone to one more FLUXNet Early Career Networks seminar. We have a couple of really great talks for you today. I'm Gilberto Pastorello, and along with Halson Chu and other members of the Early Career Network, will be helping with the questions on the background. And from Lawrence Berkeley National Laboratory, here's your host, Ling Meng. Take it away, Ling.
Hello and welcome everybody. Thanks for joining FLUXNet webinar. I'm Lin. I'll be your host today. Just a quick overview of what we can expect today. We'll have two speakers, Dr. Lianhong Gu and Dr. Jingfeng Xiao, talking about SIF and its application in this webinar. The webinar is 1 hour. Each talk is 25 to 30 minutes. And we'll have time for 1 to 2 quick questions at the end of each talk and a longer Q&A at the end. Feel free to ask questions anytime here in the chat, and we'll get them to the speaker. The session will be recorded and will be available on our YouTube channel later. Okay, I'd like to introduce our first speaker, Dr. Lianhong Gu. He is a distinguished research staff scientist in the Environmental Sciences Division, Oak Ridge National Lab. He conducts research in photosynthesis and ecosystem science with observational, theoretical, and experimental approaches. He is the inventor of the licensed technology fluorescence auto measurement equipment, the software, integrated measurement and control system for SIF, and the community online tool, LeafWeb. The title of his talk is The Light Reactions of Photosynthesis and the Grand Hypothesis of Eddy Covariance Energy Imbalance. So welcome, Dr. Gu.
Hi, everyone. Thanks to Ling and FLUXNet and AmeriFlux for inviting me to give this talk. Like many of you, the energy balance problem in EC measurements has been bothering me for more than 20 years. So the energy imbalance in eddy flux measurements is perhaps the darkest cloud hanging over the whole community.
Dr. Gu, you haven't shared your screen yet.
Oh, really? Okay, sorry about that. Do you see my screen now?
Yeah.
Okay. Okay. Sorry, everyone. Yeah. So the like many of you, the energy imbalance in EC measurement has been bothering me for 20 years, and this problem is perhaps the darkest cloud hanging over the whole EC community. If we cannot even get the energy budget closed, how could we trust our measurements? Just to refresh our mind, what the EC energy balance problem is. If you add all the flux terms and the storage terms together, the sensible heat flux, ground heat flux, latent heat flux, heat storage in the air column, latent heat storage in the air column, the biomass heat storage, the moisture biomass, and the photochemical energy storage in carbohydrates. The sum of these energy terms is only about 80 to 90% of the net radiation. This imbalance occurs essentially at every flux site in the world. So because the energy has to be conserved, a question arises: should easy flux measurements be adjusted to account for this energy imbalance? In fact, some people have done just that. Several years ago, I realized, wait a minute, we have not been considered all the energy storage terms in the ecosystems. So typically, easy community calculates half-hourly fluxes. At the half-hourly time step, energy can be stored temporarily in photochemical reaction products. Such a transient photochemical energy storage are not detectable by the easy sensors at the time of storage. But until recently, we could not test, whether, even determine the magnitude of photochemical energy storage at the ecosystem scale. But this has changed. The new advances in measurement technology and also in theoretical development has made it possible to evaluate the magnitude of photochemical energy storages at the ecosystem scale. So I decided to give this talk. So what are the transient photochemical energy storages? Photosynthesis consists of light and carbon reactions in sequence. The light reactions absorbs photons to split water. To split water, drive electron transport, and create pH gradient across the thylakoid membrane to generate ATP and NADPH as temporary energy storages. And the carbon reactions then use the ATP and NADPH generated to reduce sugar, to reduce CO2 to produce sugars. To form, these are the produced sugars essentially represent the permanent energy storage. The lighter reaction, the lighter energy harvested by chlorophylls is first stored in the following three forms of photochemical energies. One is the generation of a pH gradient across the thylakoid membrane. This is also known as proton motive force, and the production of ATP and the production of NADPH. This is the diagram of lighter reactions. Although it may look, complex, but it's actually not that difficult to understand if you just follow the arrows. Look at this diagram. So there are two types of photosystems: photosystem 2 or PS2 and photosystem 1 or PS1. To understand lighter reactions, you need to start from PS2. The two photosystems are numbered in reverse because PS2 was discovered first. And then the two photosystems are embedded in the thylakoid membrane. Also embedded in the thylakoid membrane is the cytochrome B6F complex and the ATP synthase. Both photosystems can absorb light energies at the same time. Once the PS2 reaction center chlorophyll absorbs a photon energy, that energy can either come from direct absorption of itself or from the surrounding antenna complexes. Once it absorbs a photon, red photon energy, one electron is locked off of the reaction center chlorophyll, and that electron reduces a molecule called plastoquinone. And at the same time, this energy is also used to run the oxygen evolving complex, which results in this in splitting water to produce oxygen and electrons and protons. During the as the electron produced from PS2 transport downstream, the protons also is transported from the stroma, which is the upper part, to the lumen, which is the lower part or the inner part. And as the PS1 absorbs another, another photons, one electron is locked off the PS1 reaction center chlorophyll. That electron is used to reduce NAD plus to NADPH, and that's form a linear electron transport. And then there is possibly another electron transport a pathway, which is the cyclic electron transport. In this transport route, an electron from PS1 is returned back to the plastoquinone, and then to cytochrome B6F complex, and to the plastocyanin, and finally back to PS1. That's making a circle. That circle does not produce any NADPH. However, it does help transport transport protons from the stroma to the lumen, and there will be the lumen would have higher proton concentrations than the stroma, which forms a proton gradient. This proton gradient essentially represents potential energy storage. This electrical potential energy drives the transport of protons back from the lumen to the stroma, which generates ATP. So we have three energy storage processes during the LIDAR reactions. One is the proton gradient, which is also known as a proton moving force. The two chemical high energy chemicals, NADPH and NADP. These three energy storage forms are at the core of my explanation why the EC energy balance is not closed. So let's talk about each of them in some detail. Okay, so the proton motive force is generated by the LIDAR reactions in three ways. One is the water splitting in the lumen by the oxygen evolving complex. And in this process, 4 radar electrons energy is used to split 2 water molecules to produce 1 oxygen molecule and 4 electrons and 4 protons. And then both the linear and cyclic electron transport result in proton pumping from the stroma to the lumen. And then in the stroma, 1 proton is consumed as NADPA is reduced to NADPH, which effectively makes the proton gradient and thus the PMF stronger across the dichloride membrane. The PMF, the proton motive force, is consumed as ATP is produced. So the NADPH is produced with the electron from PS1, And during this process, the 4 red photons energy is used to reduce the NADP plus two molecules of 2 NADPH. So this process requires 4 red photons energy for every 2 NADPH. 2 NADPH are consumed to assimilate 1 molecule of CO2 into carbon hydrate, and at the same time to regenerate 1 RUBP in the carbon reactions. So the ATP is produced, is synthesized by the ATP synthesis in the thylakoid membrane. And this process converts ATP and the inorganic phosphate into ATP molecules. And for 3 ATP produced, you have to move 12 protons from the lumen to the stroma. In C3 photosynthesis, 3 ATP are consumed with 2 NADPH to assimilate 1 CO2 molecule. In C4 photosynthesis, let's assume we're talking about the NADP malic enzyme subtype. You need 2 additional ATP to concentrate CO2 at the Rubisco. So C4 photosynthesis actually demands more energy. So let's do a counting. How many photons we need in order to assimilate 1 CO2 molecule under ideal conditions? The C3 photosynthesis consumes 4 red photons in PS2 at 680 nanometer, and 4 red photons in PS1 at 700 nanometer for each CO2 molecule assimilated. The C4 photosynthesis requires 4 red photons in PS2 and 8 red photons in PS1 for each CO2 assimilated. The 4 extra red photons in C4 species are needed in order to pump 8 protons from the stroma to lumen for the synthesis of 2 additional ATP needed by the CO2 concentrating mechanism. The energies of these photons are transferred to the photochemical energies contained in PMF, NADPH, and ATP. And our eddy current sensors do not know that. Okay, so how much energy storage, photochemical energy, storage we are talking about? Let's take this cotton leaf as an example. And in this example, the linear electron transport at the maximum linear electron transport at PAR of about 2000 is about 400 micromoles per meter square per second. This linear electron transport rate corresponds to 70 watts per meter square absorbed by PS1 and 680 watts per meter square absorbed by PS1. So, but I need to, you need to be aware, this amount is not necessarily the missing energy in EC energy imbalance, because under steady state conditions, a small part of this energy is stored in sugar, but the rest is simultaneously released as heat detectable. As long as it's released as heat, it is detectable by our EC sensors. However, the key words here are under steady state conditions. So, but what if the condition is non-steady state? It's not in steady state conditions. Then, if it's not in steady state conditions. The complete energy balance equation is this one. It has so many more terms than the typical equations the EC community has been dealing with. I grouped these energy terms into 3 categories. One is the physical or EC energy terms that typically we analyze. And then there's this biochemical energy storage in carbohydrates. And then there are these photochemical energy storages. Here I added the energy potential energy storage associated with the formation of triplet state of chlorophyll, which could result in the formation of reactive oxygen species. These are also high energy particles. And also you could have emission of isoprene. The emission of isoprene also represents, some energy, but I'm not going to talk about this term today, but just to be aware, there are more energy dissipation pathways other than the PMF, NADPH, and ATP. So now the problem is that our eddy current community has been analyzing the surface energy budget with this incomplete equation. And the problem is that there's no steady state under the sun from the energy balance point of view. Even under perfectly clear sky conditions, we still have this diurnal cycle in solar irradiance. On top of this diurnal cycle of solar irradiance, we have clouds, we have wind, which could create, fluctuations in solar irradiance upon a leaf, particularly if the leaf is under the canopy. So we could not, for energy balance analysis purpose, we could not assume the ecosystem is under steady state condition. Therefore, whether the EC measurements can close the surface energy budget should be analyzed with the complete non-steady state equation. Okay, as I said in the past, we could not actually evaluate, how much photochemical energy storage is there at the ecosystem level. But with the development of measurement technologies of sun-induced chlorophyll fluorescence, we could actually do that. So, but to do that, to determine the photochemical energy storage, we have to couple the SIF measurement and eddy current measurements together. So, from the SIF measurements, we could calculate the instantaneous linear electron transport rate with the theoretical equation I developed 2 years ago. You could check out this paper in New Phytologist. This equation here is actually valid for both C3 and C4 species. From the EC measurements, we could calculate the average linear electron transport rate that is demanded by the carbon reactions. Then the difference between the linear electron transport rate calculated from SIF and from EC could be used to calculate the photochemical energy storages. The fluorescence auto measurement equipment, or FAME, was designed specifically for joint measurement of SIF and eddy flux measurements. And please check out this paper in JGR by Geosciences. Okay, so now let's make a formal statement of the photochemical hypothesis of the apparent EC energy imbalance. The EC energy budgets are apparently imbalance because there are significant net storages of photochemical energy in the form of PMF, NADPH, ATP, and potentially also reactive oxygen species under highlight. Subsequently, these photochemical energy storages, because they are transient storages, so they cannot stay for long. So they dissipate or are used under low light or even in the evening times. Okay, this hypothesis have many implications and also can be used to make many predictions. Here are a few examples. The implications: the apparent EC energy imbalance is not an indication of deficiency of the EC approach. EC measurements of energy and mass fluxes, including all measurements, sensible heat flux, latent heat flux, CO2 flux, water vapor flux, etc., or methane flux, they are okay, should not be adjusted to force a closure of energy budgets. Under highlight conditions, the linear electron transport rate calculated from instantaneous Swift measurement should be larger than the average electron transport rate estimated from the eddy current measurement. The relationship between the EC energy sum, if we use it as Y, versus the native radiation, if we use it as X, has a slope less than 1. And this relationship does not go through the origin. Morning and afternoon differ in energy balance because with more missing energies in the morning than in the afternoon, because you have to build up the photochemical energy storages in the morning. So energy closure improves as averaging time increases. Energy imbalance is worse over high productivity sites than over low productivity or deserts. Energy imbalance is worse during the growing season than during the non-growing season. But be aware of a potential photochemical energy production of evergreen species in winter. Finally, land surface models should consider photochemical energy storages and their out-of-phase release as heat in order to correctly simulate land surface fluxes and temperatures. Okay, so if you believe what I have said and want to prove it, or if you do not believe what I said and want to falsify it, please contact me. Thank you.
Thank you very much, Dr. Gu. That's really a great presentation. Now we have time for 1 to 2 quick questions. If you have a question, you can go ahead and unmute yourself and ask, or you can type in the chat. We already have 2 questions in the chat. So a question from Dave. ATP and NADPH will be consumed by the plant. The PMF will dissipate in the dark. Where is the mechanism for the storage? Somehow I'm missing this, even if things are transient. This biochemical energy pool disappeared.
Yeah, so time scale is the key. So the energy is stored at the time when sensors could not see, but the energy is released during the time when the energy could be detected by EC sensors. This creates a out of sync between the net radiation and the consumption of these energy terms. Yeah, so if it's all about the time scale. Again, it's all about the time scale. If you average over 24 hours, these terms should be 0. You should get the energy balance. But then a lot of times we actually do not measure actually measure all these terms like, like, for example, we do a very poor job in ground heat flux measurement. So it's a matter of averaging time steps.
Thank you. Great. We have another question, part of which is temporally offset compared to other energy fluxes, and lack of energy balance closure is 10 to 30%. Isn't there an important role for the other processes you listed on your slide 2, ESP storage?
Okay, I could not. Okay. So are you talking about the magnitude of other energy terms instead of the PMF, NADPH, and ATP? Is this what exactly is this question about? I did not quite understand that question.
Okay, Dr. Gu, maybe we could leave this question in the end, and we can move to the second talks.
Okay, good.
Okay. So now we'll move on to our second speakers. Our next speaker is Dr. Jingfeng Xiao. He is a global ecologist at the University of New Hampshire. His research is motivated by the compelling need to understand the impact of climate change and human activities on the Earth's biosphere and the feedback to the climate. Although he has never established or maintained any flux site, he uses flux measurement for almost every project. With expertise in remote sensing and modeling, he has been upscaling flux and FLUXNet data to the continental and global scales, and has also been heavily involved in synthesis and model improvement efforts. The title of his talk is Assessing Drought Impact on Terrestrial Photosynthesis with Satellite Data from OCO2 and SMAP. Welcome, Dr. Xiao.
Thank you, Lin. Can you see my slides?
Yeah, that's great.
Okay, cool. Hi, everyone. It's my pleasure to have this opportunity to share a part of the research we have been doing over the last couple of years with all of you. First of all, I would like to thank Lin and Gilbert for inviting me and organizing the seminar series. Lianhong just gave us such a wonderful talk about the SIF. I'm going to change the gear a little bit and talk about SIF measured from space, assessing the impacts of drought on gross primary production, or GPP. We SIF from OCO2 and soil moisture and GPP data from SMAP. OCO2 and SMAP are two relatively new NASA satellites. Most of the work was done by my postdoc, Xin Li. I would also like to thank all my co-authors here. Drought could have substantial impacts on ecosystem productivity and carbon uptake at regional to global scales. Drought is an important source of the interannual variability of the terrestrial carbon budget. It is also projected to be more frequent and more severe during the remainder of the 21st century. So understanding the responses of ecosystems to drought can inform agricultural and forest management and the projections of carbon climate feedbacks. As there have been many severe and extended droughts across the globe. In 2018, as the US Southwest experienced a severe and prolonged drought, it impacted a large fraction of the region throughout the growing season, particularly the so-called Four Corners region, Utah, Colorado, Arizona, and New Mexico. Remote sensing has been traditionally used to study the responses of ecosystems to drought, in particular vegetation indices, such as the normalized difference vegetation index, or NDVI, and the enhanced vegetation index, or EVI, have been used either as proxies for plant productivity or as key inputs to models, such as light insufficiency models. Traditional remote sensing techniques have limitations for monitoring productivity, particularly in dryland-dominated regions, such as the US Southwest. Over the last decade also, there have been some emerging data and techniques. One emerging technique is solar-induced chlorophyll fluorescence, or SIF. SIF is a believed to be a really promising and strong proxy for gross primary production. Global SIF retrievals are now available from several satellite platforms or instruments such as GOSAT, GOMI2, OCO2, and CHOPANI. GOMI2 provides the longest SIF record starting from 2007. OCO2 provides SIF observations with the finest spatial resolution from space. The emergent SIF technique is becoming so promising and popular that is considered as one of the milestones in remote sensing of the terrestrial carbon cycle. This is a diagram from our review paper of the advances over 50 years in remote sensing of the terrestrial carbon cycle in remote sensing of the environment in 2019. In this study, we synergistically used SIF data from OCO2 and soil moisture and GPP product from SMAP to assess responses of ecosystem productivity to the 2018 drought. This paper was very recently published in the same journal. The OCO2 has been providing SIF observations since September 2014. The ground area of the SIF soundings is roughly 1 kilometer by 2 kilometer, which is pretty close to the footprint of many eddy covariance flux tower. This is pretty exciting because it allows for linking the SIF measurements from space with flux tower GPP directly. So a bunch of studies have used the OCO2 SIF data and flux tower data to look at the relationship between SIF and GPP at the ecosystem scale. Our own studies published in Global Change Biology showed that there is a strong relationship between SIF and GPP at the ecosystem scale for a wide variety of biomes, except evergreen broadleaf forest, where we had a very limited number of data points. More interestingly, it looks like the slope of the SIF-GPP relationship is pretty much the same across all the biomes. It appears there is a nearly universal relationship between SIF and GPP at the ecosystem scale. Although OCO2 provides the SIF data with the finest spatial resolution and more frequent observations compared to previous SIF missions, such as GOMI2 and GOSAT, there's still a lot of data gaps. The map here shows all the overpasses for a single month in 2015. As we can see, there's still a lot of data gaps across the globe. Of course, we could aggregate those individual discrete SIF soundings into a gridded data set. We could use coarse resolution grid cells, and we could aggregate the data to a relatively long period of time, for example, 2 weeks. Even if we do so, we could still have substantial data gaps here or there across the globe. So in previous studies, we used MODIS data and the meteorological data along with a machine learning approach to fill those data gaps. This way, we get spatially and temporally continuous SIF estimates for each 0.05 degree grid cell and for each 80-day interval from 2000 to 2018. Besides SIF data from OCO2, we also use data from the soil moisture active passive, or SMAP, a NASA satellite launched in January 2015. This satellite provides global soil moisture retrievals with 9 to 36 kilometer spatial resolution and favorable accuracy. We used the root zone soil moisture science data product based on SMAP. In addition to that, we also used the GPP data product from SMAP. This product is based on a MODIS GPP algorithm. On top of the MODIS GPP algorithm, it adds on another water constraint, which is based on the SMAP soil moisture. So it basically uses VBD and soil moisture as constraints or scalars to account for the atmospheric and soil moisture conditions. So we use SIF data from OCO2 and soil moisture and GPP data from SMAP. These two satellites have been collecting about 6 years of overlapping observations and provide a really nice opportunity to look at the responses of ecosystems to drought. We also use the maps from the US drought monitor to match the intensity and evolution of the drought over time. We also use the temperature, BPD, and PAR from meteorological real analysis data set and flux tile data, as well as county level crop yield. And we spatially averaged SMAP soil moisture, VPD, SMAP GBP, and SIF over the 7 states that were severely affected by the drought for each month over the growing season and for each year from 2015 to 2018. The red line indicates the 2018, the drought year. The soil moisture exhibited the lowest value over a large portion of the growing season in 2018, and the VBD had the highest value from April to June or July. So the drought suppressed plant productivity. So as a result, while both GBP and SMAP had the lowest values over a large portion of the growing season in 2018. While spatially, we could see a lot, we could see many grid cells in the southwest had negative anomalies in soil moisture and positive anomalies in VBD. These are anomalies averaged over the growing season in 2018. By the way, we use 2 color ramps here on the right, and we use the top one for GBP, soil moisture, and SIF, and the bottom one for VBD. So the majority of the grid cells in the southwest had negative anomalies in soil moisture and positive anomalies in VBD. And for those grid cells, there were large negative anomalies in GBP and SIF. And the spatial patterns of both productivity measures were pretty consistent with those of soil moisture and VBD. We could sort of decompose SIF into 2 components, the APAR and the SIF yield. There were decreases in both components, APAR and SIF yield in the southwest. So this indicates the decline in SIF in 2018 was due to the decreases in both components, APAR and SIF yield. And we also look at that PAR and FPAR. It looks like was a decrease in APAR was mainly caused by the decrease in FPAR. There was no big change in the APAR in the southwest region. This figure shows the spatial patterns in the temporal evolution of the drought as indicated by the US drought monitor maps on the left. And the right 3 columns indicates the anomalies in soil moisture, GBP, and SIF from May, July to September. This map, soil moisture product, really tracked the spatial spread and the temporal evolution of the drought fairly well, as indicated by the US drought monitor maps. And the water stress suppressed the plant productivity as indicated by both SMAP GBP and GUSIF. So the spatial patterns of the productivity measures were pretty consistent with the measures of with the patterns of the soil moisture and the drought maps, spatially and temporally. We then look at the relationships of the monthly anomalies in soil moisture with the monthly anomalies of SIF, SMAP, GBP, APAR, and SIF yield. The color indicates the correlation coefficient. Also, correlation coefficient, the values for the grid cells in the southwest region are all positive. The majority of the grid cells had pretty strong correlations between soil moisture with SIF and SMAP GBP. It appears that SIF was slightly more sensitive to the variations in soil moisture. We can look at the relationships between soil moisture and APAR, or SIF yield as well. The relationship is generally pretty weak for APAR and is pretty strong for SIF yield. These results indicate the sensitivity of SIF to the variations in soil moisture was mainly caused by the sensitivity of SIF yield to the variations of soil moisture. We were also aggregated these variables due to the regional scale as the 7 states severely affected by drought. And then look at the relationships between soil moisture anomaly and the productivity measures, as well as APAR and SIF yield. So relationships at the regional scale are pretty consistent with those at the grid cell level CIF was slightly more strongly related to soil moisture anomaly compared to SMAP GBP. And the soil moisture anomalies was strongly correlated with CIF yield, but weakly with APOL. Both SMAP GBP and GoCIF could also capture the impacts of drought on flux tower GBP. We looked at 4 sites here across the Southwest, so all affected by the drought in 2018. And the drought actually started prior to the growing season for all the 4 sites. And we could see a large decline in flux tower GBP from April to August for all the 4 sites. And both SMAP GBP and CIF could capture the decline in flux tower GBP and the seasonal variations as in response to the drought. And both productivity measures could also capture the impacts of the drought on county level crop yield. The y-axis here is the anomaly in crop yield at the county level, and the x-axis is the anomaly in CIF or SMAP GBP. We see that those counties having large soil water deficits typically had decreases in crop yield at the county level. And those counties having favorable soil moisture conditions typically had positive anomalies in crop yield. Just a brief summary. This exercise will demonstrate the potential for using OCO2 data and SMAP for studies responses of ecosystem productivity to drought. Both productivity measures responded strongly to anomalies in VBD and soil moisture. CIF was slightly more sensitive to soil water stress than SMAP GBP. There is a lot of potential to use data from the 2 satellites, OCO2 and SMAP, synergistically for studying the responses of ecosystems to drought. Well, we also developed a GBP product based on the CIF data product. We used the CIF GBP relationships we established, and then we converted the CIF to GBP for each grid cell and for each 80-day time step. Both global scale data sets are available at our data repository, data.globalecology.unh.edu. We're currently extending the data to 2020. We should be able to make 2 extra years of data available online within 1 or 2 weeks. You're most welcome to use the data sets for research. I want to briefly talk about another study that we conducted a couple of years ago. We looked at the impact of the 2015 drought on the Amazon forests. The story here is slightly different. At that time, we used CIF data from GOMI2. So the figure here shows so a large negative anomalies in precipitation, soil moisture, and then positive anomalies in BBD. But surprisingly, we did not really see widespread declines in the CIF signal. And in fact, if you look at the grid cells more carefully, many grid cells actually had increases in CIF signal, which is a little puzzling. But later we decomposed the CIF into APA and CIF yield. So CIF yield was exhibited widespread negative anomalies. And if you look at the APA, APA had positive anomalies for a large portion of the Amazon basin. And if we decompose the APA into its 2 components, PAR and FPAR, we can see there was an increase in PAR and FPAR as the 2 signals. So there was a low cloud cover during the drought period, and there was more solar radiation reaching the canopies of the forests and the savannas as well. And it's probably also because of the flushing of the new leaves, there was an increase in leaf area index during that period of time. So there was also increase in FPAR. So this is just a brief story, which is slightly different from what we found in the US Southwest region dominated by dryland ecosystems. Well, the GOMI2 dataset is really cool, but it has some problems. It's subject to sensor degradation. So you would be really careful if you want to look at the trend over a long period of time. And it also has a relatively close spatial resolution. For OCO2, it has a relatively sparse observations over space and time. If we want to get fine resolution data sets globally, we still need to bring in MODIS data or OS data sets to fill those data gaps. But now we have another data set at our disposal. This data set is from Chalpami, an instrument on board Sentinel-5 precursor. This instrument has been collecting SIF data with around 7 kilometer spatial resolution globally since December 2017. It's a really cool data set, which is really useful for a lot of regional and global scale studies. We could aggregate the Chalpami SIF soundings to a graded data set. It really depends on what kind of spatial resolution and temporal resolution you would want to use. For example, if we use the daily time step, we can get a lot of data gaps across the globe, or the continent of North America, regardless of the grid cell size we use, 0.2 degree or 0.05 degree. But if we use 8-day time step and 0.05 degree, we could have a pretty much complete coverage of the continent or the globe. So seeing very few data gaps, and we could use this data set to look at the impacts of drought on ecosystems at the regional to global scales. Well, the Chalpami data set is really cool, but its spatial resolution is still relatively coarse. So we can have like a half a degree, 0.05 degree, or 7 kilometer spatial resolution, and like 8-day time step, but it's still relatively coarse, especially for heterogeneous regions. For example, the dryland dominated regions like the US Southwest. The good news is, well, there will be a new satellite called FLUX. It will be launched in 2022 or maybe even later. It will provide global SIF observations with 300-meter resolution. So this data set will be really exciting for a lot of studies. Another really cool data set is from the OCO3. Unlike its predecessor, OCO2, OCO3 is on board. It's not on Apollo orbit. It was deployed onto the International Space Station in 2019. Because of the special unique orbit of the International Space Station, the platform or the instrument has diurnal sampling capability. So we could get SIF observations for different times of the day. This is just one figure, as one example. The map on the top shows SIF for the early morning, around 9 o'clock this morning for January 14, 2020. And the map at the bottom is SIF acquired in the afternoon, around 4 o'clock, 4:30 on the previous day. This area is in southeastern Australia. So we can see, well, in the early morning, the photosynthesis rate was pretty strong. Many plants had high SIF values at that time of the day. But around 4:30 in the afternoon, many plants had fairly low SIF values, indicating those plants have fairly low photosynthetic rates. This is likely because of the water and heat stress at that time of the day and the subsequent stomata closure. So the SIF data set from the OCO3 is really exciting, which can potentially allow us to look at how photosynthesis changes over the course of the day in response to water stress, heat stress, and other environmental or physiological factors. With that, I would like to thank the support of NASA and the National Science Foundation, as well as the support of AmeriFlux, FLUXNet-PIs, and OCO2, OCO3, MODIS map, and Chalpami science teams. Thank you.
Thank you for the presentation, Dr. Xiao. That's a great talk. Now is our Q&A session. We already have some interesting discussion in the chat. If you have questions, you can go ahead and unmute yourself and ask, or you can type in the chat. There is a question to Dr. Xiao. Could you comment on flash drought affecting forest productivity in the wet region in the US? How good is this map for forest conditions?
That's a good question. Well, I think we'll see if he's able to do a pretty good job in the forest regions, for example, in the eastern United States. We did not really look at the impacts of drought on those regions per se, but some interesting results are included here. For example, if you look at the slide here, we could actually see anomalies in surf and SMAP GVP in the eastern United States and or in the New England region as well, or in the Midwest. But those negative anomalies in both productivity measures were caused by different factors, as were not really caused by drought. We decomposed the signal to APAR and yield, and so we also look at some PAR data, FPAR. It looks like, well, in those regions, the declines in GPP and surf were mainly caused by some lower solar radiation.
That's great, thank you. Other questions? Dr. Gu, there is a question that came earlier, I think, for you. Did you ever quantify the transit photochemical term? How do they correlate with the measured imbalance?
Hello, can you hear me?
Yeah.
This is we are still working on the data analysis part. So as you can tell from the title of my presentation, it's a hypothesis, and it takes you know, as I said, I have been bothered by this problem for 20 years. I could wait for a few more years to get the data ready and get the methodology ready. But just to be clear, before we do any examination of the photochemical energy storage terms, we have to get all those easy terms right. We have to measure the heat storage in the canopy, heat storage, and we have to measure the ground heat fluxes correctly. These are the easy terms. We need to evaluate, get the easy terms right first. But if you look at all those, you know, the energy balance analyses, I mean, you know, not all those papers considered the easy terms, you know, very thoroughly. So just another point I want to clear this: the energy imbalance in eddy covariance measurements is apparent, is a scale mismatch problem, is the out of phase between the net radiation and those dissipation terms. In other words, we don't have to worry about the eddy covariance technique itself. And someone commented about the WPL correction term. This has nothing to do with our, you know, the eddy covariance, the photochemical energy storage term I'm talking about. The WPL is a physical requirement. It must be applied.
May I ask one question?
Yeah, sure.
Yeah. So, Lianhong, I'm so excited to listen to your talk. Is you put a budget balance equation with some new terms, ATP, NADPH, and something else. I'm so excited about that idea, and I'm also worried about all this biochemical energy can be measured by this just dry matter, organic matter. There's a biochemical energy there. You have one term 0.48 something time. I think that should be dry matter. And I worry about so in your part of the equation, so something like ATP, NADPH, you counted 2 times, or so you see energy conservation, right? Yeah.
Well, first of all, the that 0.48 times NECO2, that is what you are talking about, the biomass energies, chemical bond energy storage. In other words, that's the energy stored in the carbohydrates. This is not the photochemical energy I'm talking about. The photochemical energy storages are transient, but they are not like or they store at at the 10 o'clock and then get consumed at the 10 o'clock. Then there would be no problem about they would not contribute to the energy imbalance.
So
right?
So this biochemical energy, ATP, NADPH, if not used, that would be waste. That would be not being storage in the biochemical energy. That's glucose, right? So
that's correct. That's correct.
That means just spontaneously energy, just like a solar energy. You got a solar energy panel, you got it, but you do not store that energy. That energy will be gone. So that it would be just like bar. It's just get out of ecosystem, right?
That's why I have emphasized the energy balance problem is really a time averaging issue. It's a time scale issue. It's an out of phase problem. So it's like you accumulate some energy in 10 o'clock, and then it gets consumed in 5 p.m. Something like that. So it's really about the phase relationship between the incoming energy and the energy consumption. Yeah, so it's a scale problem.
Lianhong, if you follow your idea, then your energy measurement, you're supposed to have sometimes lower, sometimes higher.
Exactly.
And then so, why this systematically be lower by 10 to 30%?
Well, if you look at the relationship between all those traditional energy terms, the sum against the native radiation, they don't go through origin. Okay, so in the my prediction is if you separate the morning and afternoon time, that intercept would be negative in the morning and positive in the afternoon. Okay, so that energy gets consumed, stored under high light and consumed under low light. But then again, we have to be careful. We have to measure those traditional terms correctly. We have to do the labor to measure them before we evaluate the photochemical energy terms.
Anyway, I love your spark idea. That's exciting for us. But yeah.
Great. Thank you for the discussion. We have a couple of questions coming direct to Dr. Xiao. The first question is, could you comment on the biodependence of the GPP-CIF relationship?
Oh, that's a pretty good question. Well, according to our analysis, it appears there's almost a universal relationship at the biome level. But there's one exception, which is the evergreen broadleaf forest. We only had a very limited number of data points, and those data points together only account for a small fraction of the growing season. So it basically only accounts for a small part of the seasonality. So but now, while we have a lot more years of data available, so if we revisit the relationship based on OCO2 data and the flux tau data, that relationship might be different. Another point is also the relationship seems to be universal and across the biomes, but as relationship does change with for the synthetic pathway, it could be quite different for C3 versus C4 ecosystems.
Great, thank you. Another question is, does your OCO3 paper and review address diurnal Sif variability, diurnal Sif LST relationship?
The answer is no. We analyzed those data was simply to provide example to demonstrate the OCO3 instrument has a diurnal sampling capability, and it has the potential to allow us to look at how photosynthesis changes over the course of the diurnal cycle. So we did not really go that far, yeah, but that would be something interesting to pursue in the near future.
Great, thanks. And the third question is about the Sif yield. A number of studies have indicated the importance of canopy structure in satellite Sif. So based on your calculation, this canopy structure information should be embedded in the Sif yield part. Do you have any ideas of how much Sif yield is caused by structure changes and how much is caused by leaf level fluorescence yield change?
That's a really nice question. It involves the canopy structure and a more process level based understanding. Well, we did not really go very far at this point, but I guess, well, the canopy structure information is actually embedded in both components, both Sif yield and APAR, because what APAR is a product of PAR and FPAR. So FPAR could partly account for the canopy structure information, and for Sif yield, the structure information is probably comes from the escape ratio of Sif. So it's more complex the canopy structure, maybe the lower the escape ratio. So if we really want to look at that canopy structure information, we probably need to bring in additional data sets or maybe to do some corrections. That would be something interesting to pursue.
Great, thank you. And go back to Dr. Gu, we have a question in terms of the time scales. So what's the time scale of the photochemical energy storage? EC energy budget is useful averaged at half-hour time scale.
It would depend on the actual environmental conditions. It's hard to determine. So if there's a continuous energy accumulation, then the PMF and NADPH and ATP are going to accumulate. So they are not going to get consumed. But then there would be a problem. The plants might be damaged, due to the generation of reactive oxygen species. It's hard to predict. But I would say, at the diurnal time scales, we probably could average out the photochemical energy storages. But then again, all those traditional storage terms have to be measured carefully.
Great, thank you. And a follow-up question on that is a weaker correlation between GPP and Sif at a smaller time scale could be related to the storage component that is non-zero at small time scales.
Yeah, so I answered that question. So even if under steady state condition, there's no storage of photochemical energy, the relationship between Sif and GPP would be still complicated. because there are so many factors that can affect that relationship, biological factors and environmental factors. And when we evaluate the CFGPP relationship, we have to use the theoretical equations we have developed. So we cannot just say, oh, we can coordinate, CF and GPP, but then it's a coordination. But then when you add the non-steady state conditions, the relationship would be even more complex. So it's what I would suggest, we have to carefully look at the theoretical models, in order to evaluate this CFGPP relationship.
Great, thank you. Okay, I think that covers all the questions we received. I just want to thank both of our speakers one more time. It's really great presentations. And thank you everyone for joining us again. This recorded session will be available on our YouTube channel later. Also, you can find us on Twitter, FLUXNET-ECN, and sign up for our emailing list. Okay, be safe and we'll see you at the next seminar. Thank you.
Thank you for having me, Lin.
Great, thank you.